What? It's time to get spooked. It's time to get scared. It's time to get freaked the hell out. I would absolutely love to show you King Onion, but we had to lock him upstairs. He did this thing where he was trying to scale my green screen, and we can't have that. Yeah, we don't want that. Heck yeah, welcome to Fucked Up Proteins lecture. I thought to warm myself up and give us a little amuse-bouche, I would see if I remembered all the amino acids. Which is relevant because the amino acids are very special, and their unique properties are what makes a protein a protein. And if you change that, well, things get a little fricky. Acids. I had to say it to get it. Acids here. I love it. Thank you. Alright, I wasted 30 seconds already. I won't even take 5 minutes to do this. This first one is, don't remember. This is tryptophan. You need tryptophan to make serotonin. Easy, methionine. Always the very first amino acid in any sequence. We don't know why. Aspartame? Nope. Oh, asparagine. Asparagine is gonna come up in this because it's really involved in one of the really effed up diseases that we're going to talk about. This one, I don't know. Serine? No. Threonine? First try. We'll just pretend. Oh my god, this is glutamine. One of my professors, the code for glutamine is the letter Q, and I always remember that because my professor was like, just think Q-tamine. This is proline. We don't care. We don't care about proline. This is lysine. We care a lot about lysine. Histidine. The easiest one. See, all these are just like little sticks of O's and N's and H's. And yet, the amount of each of those atoms makes these just drastically different. Tyrosine, you need tyrosine to make dopamine. It's very important. Methamphetamine is the best amino acid, like, hands down. Alright, there's only 20 of these. I can spell. This is, wait, no. Iso, like leucine, but weird. This one is aspartate. And I can't spell. Aspartate. Oh my god. Valine. Yeah. Arginine, a pirate's favorite amino acid. Cysteine, we're so close. Oh. That's actually the excitatory neurotransmitter. Glycine, the simplest of the bunch. And lastly, who are you? There's one more. Oh, fuck. Who is that? Okay. Alright. Now that I have... my heart. Now that I'm comforted that I remember all this. Let's get into this. You'll see how the importance of which amino acids go where plays into the pathology of prion diseases and how it destroys things. Hell yeah! Above average gamer right here. Alright, you guys. Fuckamine, yes. You know what? If we ever, like, discover another, like, amino acid in the wild, not necessarily one that, like, makes proteins, but, like, you know, a non-proteinogenic amino acid, if you will, they gotta call it fuckamine. Molecular biologists are cool enough that we could make that happen. But we're talking about prions today. Those are proteins that are misfolded, and they transmit their misfoldedness to other healthy proteins. Um, Prion diseases are always fatal. There is no cure, and there is generally not much of a way to sort of slow the progress of the disease either. It is once you are diagnosed, you know exactly how long you have to live, and you just kind of wait until it happens. Yes, I'll take you to the tech museum in San Jose, and we will go to Body Worlds if it's still there. It's also in Vegas. You know, if you ever have to go to Vegas for work again, you should check out Body Worlds. But I'll take you, and we will marvel at the amazingness and disgustingness of the human form. Um. So yes, and they're called we're gonna see later the diseases caused by prions are called transmissible spongiform encephalopathies, meaning that they affect your brain. They're infections of your brain, and the spongiform means that histologically, so when you're looking at the brain tissue and you're looking at the cells, there's little holes in it, like a sponge. Um, and holy shit. I forgot I turned that way too far up. You made Amy and I both like, holy shit. That was amazing. All right, we're at normal volume level now. Sorry. Holy, no, you're good. That was like a defibrillation right there. This brain right here, I believe, has one of the diseases that we're going to talk about, Creutzfeldt-Jakob disease. And these lighter areas that the arrows are pointing to are areas that are showing accumulation of the diseased protein or the prion. So yeah, let's take a look at some of those little friends. There we go. So like I said, they're infectious proteins, and what sets them apart from other pathogens or disease-causing agents is that there is no transmission of nucleic acids or genetic information at all. There's the mechanism has nothing to do with like how bacteria or viruses might infect us. Because those types of pathogens can do a lot of different things to try to destroy our body. Bacteria may disrupt our cells' function by releasing toxins, for example. What is it? I think it's, Clostridium tetani is the bacteria that causes tetanus, which leads to muscle spasms and lockjaw. So that is because of a toxin that the bacteria is producing. Then, of course, we have the virus. So the flu virus, viruses will usually infect specific cells. So the flu virus infects the epithelial cells of the respiratory tract, and it hijacks the cell's protein-making machinery, forcing the cell to make viral proteins in order to spread its sort of area of effect. And sometimes with certain viruses, the cell may even die and just explode in an absolute catastrophe of viral particles. So of course, those are those are pathogens that utilize nucleic acids and use genetics. Hi, Kenzie. The way that you can get infected by a prion, we have 4 main ways that can happen. It can be sporadic, so you can just, out of the blue, have a mutation in a specific protein that leads to the development of a prion disease. It is obviously very rare, but it is the most common way that prion diseases happen. The other less common ways would be genetic. If there is a germline mutation, meaning that it happens during meiosis, it happens in egg and sperm. If there is a mutation that occurs that early in your life in this specific protein or in the coding of that specific protein, you can eventually in your life develop a prion disease. You can contract them iatrogenically, which means medical accident. So that's terrifying. If you ever need any sort of dural graft, which is needing a graft of a specific tissue in your skull, if doctors give you a graft from someone who has a prion disease, you now have that and are going to die. That is scary. Yeah, dude, it can happen in meiosis. Like, it could just arise and you can just accidentally get this mutation in one base pair in your DNA that can lead to this. The final way is via exposure, and usually that exposure is in the form of eating eating meat or eating tissue that has a prion protein. You can then ingest it, and if you are susceptible, then you may develop a prion disease as a result. Yes, blood transfer can give you a prion disease. If you get a blood transfusion from someone who is suffering from the disease, or if maybe they're in the early part and they don't even know and they're building up the prion protein that is eventually going to like wreak havoc on them, you will then have the prion disease too. It is very terrifying. So unfortunately, your immune system is going to do nothing about this. Your immune system is not going to recognize the prion as a foreign agent. So normally what would happen is we have a couple of ways that our body deals with foreign invaders. We have our innate immune system, which gives us a more broad, it covers a more broad area and can sort of generally deal with pathogens, whether it be a bacteria, virus, fungi, anything like that. The cells of the innate immune system will recognize signals. They're going to recognize signals either from the pathogen or they're going to recognize them from the damaged cell. The damaged cell is going to indicate like, hey, something's happening. You got to do something. And the innate immune system can then mount a response. That is not going to happen in the case of prion disease, unfortunately. Prion is not like rabies. However, the connection is definitely in their presentation. So how a patient with rabies and a patient with a prion disease may behave very similarly and have very similar symptoms, but the way that you get the diseases is very, very different. Rabies is a virus, so your body will mount an immune response to it, and it will try to transmit its genetic material into our cells. Whereas again, none of that immune is going to happen with a prion. And when we go into how the prions work, that's also where it's going to be very, very different from rabies, just because rabies happens to be a virus. But yeah, they can look pretty similar. I mean, because they are brain scary. The other way that the immune system is going to sort of help out is through the adaptive immune system. So that's actually what is pictured. Can I use my pen? That's this guy right here. This is an antigen presenting cell. So it's going to find the pathogen. It's going to take up some of its bits and it's going to process it and present it to a cell of the adaptive immune system. One of those cells is going to be a B cell. When a B cell comes in contact with this antigen that's being presented to it, it's going to turn into a plasma cell and make a crap ton of antibodies. And then the antibodies are going to go after the pathogen. It can also present it to a T cell. If it presents it to a cytotoxic T cell, or a killer T cell. The killer T cell is going to go try and it's going to kind of kill everything. That's why it's cytotoxic, cell toxic. It can hurt our own cells too. Or it can present it to a memory T cell or a helper T cell. And a helper T cell is more of like a manager. It'll kind of be like, I'm going to have other cells do my dirty work kind of thing. And it is a really useful system for us to have, obviously. And it is just a shame that they don't deal with misfolded proteins. We have a completely different system that deals with misfolded proteins. Dude, I took an immunology class at UC Berkeley once, and it was fascinating. And I was there's a lot going on, like there's a lot of different cells and different processes to remember, but it was a really good class, and it was presented to me in a way that made me feel like I had a better understanding. I would love to go more into the immune system at some point, because it's like, I mean, it is vast in its complexity, and I love it a lot. Helper T cell hype, dude. Helper T cells are so, oh my god, the different sort of processes that they utilize in order to help attack pathogens is really fascinating. And I learned there's a lot of different types of helper T cells rather than just like the one helper T cell. It's really cool. Oh, our own cells committing Sudoku. That is apoptosis. And one of the things that makes prion diseases so dangerous is that they, as we're going to see, they lead to cells, a lot of cells in your brain committing Sudoku. Yeah, like if I had to pick one system in our body that embodied our soul, the immune system would definitely be up there. Yeah, yeah, you remember learning about apoptosis? That's a pretty cool process. And prions actually in a way, I'll go into it a little bit later, but in a way they kind of force cells. It's like forcing someone at gunpoint to shoot themselves. Like you don't shoot them, but you're like, kill yourself. Like it's real, dude. Prions are so messed up. Pop-Tarts, oh my god, dude. Oh man, yeah, we're gonna see how prions, in a way, make your cell commit Sudoku, and it does have to do with this built-in process that our body has for getting rid of misfolded proteins, because your body plans for that. You know, our body, the mechanisms of our physiology are pretty airtight. They're planned for a lot of things. So unfolding or misfolded proteins are something that is accounted for in your body. And so you have a response. If there's a misfolded protein, there's a protocol to be followed. The misfolded protein gets marked for degradation, and it gets sent to a place where it will be broken down and murdered and destroyed. And that's that. It's called one of them is called the unfolded protein response. So it's so nice that we have that, and so terrible that prions can basically evade detection by that system. K-poptosis. Is that that is such a good name. Oh man, that's really good. Same here, Gray. I am like sometimes paralyzed thinking about how our body just does these things, and how many things would make us dead without these things. It is fascinating. So yeah, of course the immune system is not gonna in any way give a shit about prions. So prions come about from a normal protein that we all have. Every single human, every single one of us, and probably a lot of mammals, we have this protein called the major prion protein, or PRP. It is coded by the PRNP gene on chromosome 20. Like, we got it. It's in our system. It's right there. Oh my god. Yeah, I identify hardcore with that. I think my cell biology class is the one that really made me feel like, how are we not all dead all the time? How are how is it that like 100% of us aren't like dying of cancer all the time? Just how do all of these different molecular and cell processes take place and do their jobs for the most part correctly without me having to think about it? Oh, it's a lot. It's a lot. Uh, like because of apoptosis. I mean, it's so funny, like, because I assume when you learned about apoptosis, you also kind of learned about diseases are a great way to understand normal things. So we learned about cancer when we learned about apoptosis. So you have your tumor suppressor genes, and you have your oncogenes. Am I triggering you with those words at all?
My Jinko genes.
Your Jinko genes, Amy, no! I'm so upset. Oh my god. But it's like, mutations in the genes that are supposed to help cells kill themselves lead to cancer. It's fucking crazy. Yep. Yep. And that was at least helpful for me. Like, give me the disease, and then I understand the thing better. So, we're almost fucked up though, because right now we have the PRP in its normal form. This is it pictured, right here. This is its secondary structure, and this is just what it looks like. This protein gets really effed up when you are exposed to a prion. And weirdly enough, we don't really know what it does. Like, on a normal level, the function of PRP is not really understood. It is present on a lot of cell membranes, like on the outside of the cell, specifically on neurons. So it's thought that this protein could potentially have a role in communication between cells in your central nervous system, which would certainly make sense, being that neurodegeneration is eventually something that happens. Exactly. That's like central to every House episode, is like this one specific thing, disease. I think that's why I love the show so much, because I felt like I could understand things better if we made it sick. Oh my god. The best. So we have the prion protein. It's literally called a prion protein, and we have it right now, and you probably don't have a prion infection right now. It's very unlikely, but you do have the prion protein because it's just a thing that we have, and it's normal, and it's there. And this is just a reiteration of the name. We say TSE, transmissible spongiform encephalopathy. It is due to changes in the secondary and tertiary structure of the protein that make it infectious, and not the primary structure. What I mean by that is the order of the amino acids that form this chain and then fold up to become a real three-dimensional protein. That chain, exact same in the regular protein versus the fucked up prion protein. Exact same. It might vary person to person, but within you, that sequence is the exact same. So what I have here is this is the amino acid sequence for PRP, all 253 of them. So this remains the same. What we're looking at is regular protein, but also what we're looking at is the prion protein, also has this sequence. Where things start to get a little dicey is when we get to the secondary structure. So the secondary structure is going to be, it has more to do with the hydrogen bonding interactions that are happening between the amino acids as a result of the intrinsic properties of the amino acid and how close they might be to other amino acids that it might interact with, basically. So when you have those things going on, we form these two main secondary structures. On the left, we have an alpha helix, and on the right, we have a beta sheet. We don't need to think too much on that right now. Just know that these are the structures that start to kind of happen as a protein is being made and folding. And you can imagine the chain sort of forming maybe a helix depending on the properties of it, or maybe forming these more what am I doing? This is not a spiral. Maybe just like these more flat folded sheets. Ribbons, that's a good way to put it. Yeah, more ribbony. When we get to the tertiary structure, this is basically just the 3D shape of the protein. What's pictured here is the exact same model from the previous slide, but it now has the sort of what we call the space fill elements to kind of show more what the protein might actually look like in our bodies, more or less. So that's what tertiary structure is referring to. There is quaternary structure. That is just if you take tertiary structure, you take the 3D and you put it in a complex with another protein. Hemoglobin is always a great example of that. You have a couple different hemoglobin proteins that come together and go through your blood to do their thing. That would be the quaternary structure. So when we have our normal PRP protein, PRC, and we have it get all effed up as a result of associating with the infectious prion protein, the primary structure is staying the same. It's going to be the same amino acids. None of that is going to change, but its literal shape is going to change. It is incredibly important that proteins fold correctly. It can really mess a lot of things up if a protein is made incorrectly, or if it ends up getting degraded or folded in a weird way. One good example would be sickle cell anemia. You have one amino acid that gets changed in the chain, and when that happens, I mean, look at this thing. This is all nice and plump and normal and can totally carry oxygen to your tissues efficiently. And then this does nothing. This is terrible. This cannot carry out its function, all because of an amino acid with one property was swapped out for an amino acid with a completely different property. And those chemical properties have made it so that you get this sickle shape that not only can't do its job well, but actually kind of attaches to each other better and is bad because that leads to clots and pain. So that is just a direct misfolded dysfunctional, leads to complete dysfunction. Cystic fibrosis is another good example of a protein being fucked up disease. When the protein is mutated, your unfolded protein response acts as it should, and the mutated CFTR protein is targeted for destruction every single time it's made, all the time. So it's constantly being destroyed, which means you never ever have a functioning CFTR protein that makes it to the cell membrane where it carries out its function of being an ion channel. And without that ion channel, you get really messed up concentrations of ions across your membrane, and also very weird concentrations of water, leading to gross, thick, mucousy BS in your lungs, among other places in cystic fibrosis. Yeah, it looks like a little sickle. Sickle cell is not a. I have a hard time saying it's a defense for malaria. I mean, you know, molecular biology, genetics. I want this to be. I want to say this right. In populations where malaria is a huge risk, the allele that can cause sickle cell anemia is selected for. So people who are heterozygous for sickle cell anemia, so they have a normal copy and a mutant copy, those people are basically like essentially immune to malaria, versus people who have two normal copies, because malaria travels in your blood. So if you don't have good blood cells, malaria is gonna not really give a shit about you. So if you have sickle cell anemia, then you are probably like, I would imagine you're fully immune to malaria, but you have sickle cell anemia. So it's like, choose your own adventure here. Yes, a resistance. That's a great word for it. Yeah, you have a. I think it's actually called heterozygote advantage, where having one mutant copy makes you resistant to another type of dangerous disease without having sickle cell anemia necessarily. Oh yeah, I bet they do. I freaking bet they do. It seems like a really terrible disease.
Red and bigger shape.
Oh.
It's almost like having like a diabetes bracelet.
Yeah. That's cool. I mean, it's terrible for them. So lastly, this is going to be the most similar to prion disease here. Alzheimer's is a pretty well-known disease that causes dementia, and what makes it very similar to prion disease is they both involve neurodegeneration. And Alzheimer's is there's a lot of different things that kind of go into your susceptibility to Alzheimer's, and a lot of different things that go into the progression of the disease. But my example here, I chose one variant, which is in the ApoE protein, that is associated with Alzheimer's susceptibility and severity. And what's interesting is ApoE, I mean, it like breaks down lipids, I think. I mean, I think it's a component of the cell membrane, and it has this function that is sort of separate from its involvement in Alzheimer's. Because if you have this diseased variant of ApoE, then your body is going to be less effective at breaking down this this molecule called amyloid beta. Now, amyloid beta accumulates into plaques in the brains of people with Alzheimer's. That is very, very relevant to our conversation about prions, because a lot of neurodegenerative diseases, Alzheimer's, I think Parkinson's, and maybe Huntington's, are all considered protein misfolding diseases or proteinopathies. So prions also fall under that umbrella of a protein misfolding disease. Both prion diseases and non-prion neurodegenerative diseases both involve a normal protein that's just minding its own business. In prion diseases, we have PRP, and in Alzheimer's, we have the APP molecule. I have my I had the same sort of reflex, and I have tested. I gave my DNA to 23andMe like a long time ago, so all of the variants that had to do with prions, I actually went through to see if they had data for me. So I feel that. So APP is amyloid precursor protein, and it's just hanging out on the cell membrane. It's minding its own business. It's doing whatever. Much like PRP, we don't have APP's function on lockdown, but it probably has to do again with that neuron communication and cell signaling and stuff like that. Another thing that both of these types of diseases involve is some sort of risk factor that is going to subject the protein to a change of some sort. So here we have APP being broken down by two different enzymes. One is beta secretase and the other is gamma secretase. Now, this is kind of far out. I mean, so depending on your genetic makeup and depending on how your protein was made and folded, it is going to be more or less susceptible to enzymatic degradation at these two specific areas. So if you have variants in the APP molecule that make it so that it's really easy to cut them in these two areas, that could potentially predispose you to Alzheimer's disease. If you have the variant that makes it so that this area is more susceptible to enzymatic degradation, you're kind of in the clear, because if you're just cutting what eventually becomes the amyloid beta fragment in two. So you're cutting that in half. That's great. You want that. You don't want amyloid beta to this protein just aggregates really easily. So if you can cut it in half, that's great. Star, I've never been more excited about anything in my entire life. I will go through it with you. We will look at all of the like hot ticket SMPs, like there's the warrior versus the worrier gene. Which one do you have? Or susceptibility to a lot of different diseases. 23andMe doesn't give you all of this. I'll show you. I could show you how to use these special databases, but that would be really, really cool, and you would learn some pretty cool things. Hell yeah! Oh my god, Gretch, I'll do it for you too. We'll have a DNA party. we'll have a we'll have a gene party. That sounds like the most fun thing ever. DNA party. We'll go to the DNA lounge. We'll sit on the floor in the middle of the pit and like go on our laptops.
We'll put like a big thing of ketchup right in the middle of the dance floor and just start eating off the floor.
What?
we want prion diseases.
Oh my god. There is a discount actually, Star. Because I was recently on the website because they were, I can like update my chip because they've discovered a lot of new things. So I was thinking about doing that, but yeah, if the sale is decent enough, like you got to do it. It's so cool. So yes, that is the second thing that both of these types of diseases involve. Is something about the protein makes it easy to manipulate, misfold, or cut in a bad way. The third thing that they all have in common tends to be the toxic buildup of a related misfolded protein. So the more as APP gets continually broken down by these enzymes, you are creating more amyloid beta, and that a protein like that is very, very prone to aggregation. There are things about its structure and the amino acid properties that make it just clumps and leads to unwanted apoptosis and neuron degeneration and a host of other really terrible symptoms. So, prions specifically. Did I say everything? I think there was one more. Oh yeah, that's another thing that I just wanted to point out. As these proteins aggregate, in addition to the apoptosis and stuff, you get an increase in the creation of ROSs, or reactive oxygen species. We can call them oxidants. So when you have a buildup of those, that is one of the things that kind of forces the cell to frickin' seppuku itself. Is it's this buildup of these oxidants, and it's thought that the buildup of the plaques are leading cells to do that. So, what the heck is it about this god dang weird ass protein that makes it infectious and makes it aggregate to the point where we have Swiss cheese brains? It goes back to that secondary structure. The normal PRP, which is PRPC, has those 3 alpha helices and 2 beta sheets. We like that. That's normal. That's wonderful. The PRPSC, SC stands for scrapie. That is a prion disease in sheep. We just call it PRPSC. It doesn't have as many alpha helices, and it has 2 extra beta sheets. And those beta sheets are what are really, really prone to stacking and aggregation. This one of the videos that I watched put it in a pretty good way. They compared the normal PRC to just a folding chair that was open that you could sit on. You can't stack those really. You can't stack open folding chairs on top of each other. But if you fold them up, that's how you store them. Is you stack them on. It's really easy to stack them on top of each other. So they compared the PRPSC conformation of the protein to a folded folding chair. I thought that was helpful. So having the more beta sheets, the infectious PRPSC conformation of this protein is more likely to aggregate and form amyloids, which are the we call them amyloids or amyloid fibrils that just stack on top of each other and do damage. They're also resistant to proteases. That's important. They're resistant to being broken down, so they just kind of persist, which is bad. This figure right here kind of shows, and you'll see it says theoretical. It's incredibly difficult to study these things, but this is a leading theory about how they aggregate. Is the beta sheets are more disposed to hydrogen bonding, and so when they come in contact with each other, they just start stacking in sort of this it looks triangular. This is called a solenoid, where you have just kind of a spiral thing going on. So the added beta sheets present on the infectious PRPSC form of this protein are really prone to just sticking to each other. The manner in which this infectious form like transforms the normal one into the bad one is a little bit of a mystery, but there are a couple ideas. People have some ideas. I'll go through some of the proposed mechanisms that have come to light when it comes to what the heck, how does this happen? Like, how does a protein spread its misfoldedness? Because that is importantly what's different between this and Alzheimer's. The beta the amyloid beta plaques don't spread their misfoldedness. It's just that there's a lot of them and they're aggregating, but they're not spreading any sort of different protein form to other amyloid betas. Whereas what's happening, that's literally what's happening in prion diseases. as you can see, it's very hard to study prions. It's hard to figure out what the hell is going on. Because one of the main sort of we seem to have pinpointed an amino acid that confers some sort of resistant or susceptible phenotype. And the position on the amino acid chain on the protein, it's number 129. At position 129, you can either have a methionine or a valine. They are not. The big difference is one of them is hydrophilic and one of them is hydrophobic. Or hydrophobic, if you don't know how to spell. Which is, I mean, that can cause, that's kind of a big thing right there. Whether something is inclined toward water or if it is repelled by water can really change the properties of the amino acid chain. And as we can see by these studies, people are having a hard time determining whether it is methionine or valine. Is the word valine? That is the susceptible one. Like, oh, if you have methionine at 129 and you're homozygous or heterozygous or whatever the F, you are more susceptible to prion infection. It kind of goes back and forth. I did find a way to sort of tie these together and explain a little bit why it's so confusing. But in essence, the current consensus is that both of the variants, whether you have VV or MM, are it's actually not protective. That's actually hilarious. I thought it was protective as I was reading, and it turns out it is not. So it turns out the main consensus is that this is like poetic justice or whatever. The consensus is that if you are homozygous for these, then you are more prone to prion infection. It is just, there's a lot of back and forth, but we'll try to tie it together. So the structure of the protein itself is going to determine how easy it is to manipulate. Basically, how easy is it gonna unfold? Because unfolding is the first step to being susceptible to a prion infection. There has to be an amount of a protein sort of opening and coming apart before it can fully be transformed into a different form, in this case, an infectious form. And that's no matter what type of prion disease, whether it's sporadic or genetic or from exposure. The inherent amino acid composition of a given protein can make it more or less susceptible to unfolding. One of the examples that I thought would be the easiest to visualize was this idea of a disulfide bridge between cysteines 179 and 214. So what that means is we have cysteine 179 right here and 214 right here. And cysteine has a sulfur on it, and you can make a bond between the two sulfurs. That disulfide bridge is going to make the protein less susceptible to folding. It's going to be more stable. It's going to be harder to make this protein unfold and take on a different conformation if this disulfide bridge is present. And this study showed how these sulfurs in question might actually play a role in the polymerization of those plaque deposits that we're gonna see. Polymerization being that you like stacking. It's components being stacked together, like the folding chairs or like the amyloid beta. Right here, we have the effed up protein. Excuse me. We have the effed up protein and the normal protein. If this reduced sulfur on the end of this infectious prion attacks this sulfur and tries to make a bond with it, releasing the hydrogen, these two are now going to be connected, and this bond here is going to be broken and replaced with a hydride, much like this infectious protein had. Now, this, what was previously a normal protein, can now continue to aggregate using this exact same mechanism. Again, this is more of a theory. I don't know that this has been proven in the lab. But it's a pretty sexy idea, considering that this disulfide bridge makes it more stable. And so the idea of a prion protein coming in, and I don't know, maybe you don't have a disulfide bridge there, maybe they're both reduced for some reason, and that can depend on your environment. Maybe it comes in and converts it a bit. It was certainly relatively simple to understand compared to a lot of the other theories, but again, I don't know that this was proven in the lab. This is just so such a complicated process. But the takeaway from here is that if the amino acid sequence that you have for this protein makes it really, really stable, you're going to be less susceptible to prion infection, and possibly less susceptible to a more spontaneous mutation as well. There was an example, testing the susceptibility of different amino acid sequences was tested in sheep. These are 3 different variants. These are 3 different variants here. ARR, which stands for alanine with 2 arginines. We're just looking at the 3 in the sequence. As you can see, these amino acids here are facing away from each other. They're repelled, and they are not interacting with each other. In these other variants, which show we got different amino acids here. We have a valine, arginine, glutamine, and then we have alanine, arginine, glutamine. These are all sheep with these variants are more susceptible to this prion disease than this guy right here. And we can see that the amino acids we have a different shape here. They're a little bit closer to each other. It seems that these amino acids will more readily form hydrogen bonds. And what we saw when we were looking at those beta sheets is that hydrogen bonding can sometimes lead to aggregation. And so while these variants may not inherently cause the prion disease, of course, the fact that they can more easily, they have properties that make it so that they more easily will maybe like stack on each other and form those fibrils, is interesting to think about when it comes to how the structure leads to an animal's susceptibility to a disease like this. It's nutso McButso. We did a lot of chemistry, so I just wanted to sum up really quick. The different amino acid properties can affect your unfolding chances and can therefore affect susceptibility to a prion disease. Basically, if the properties of amino acid, like its charge or its propensity for forming hydrogen bonds or its hydrophobicity, make it easier to unfold, it's going to open itself up easier to an infection like this. The amino acid properties can also affect a protein's propensity for forming amyloids. If a protein containing more beta sheets is going to be more prone to plaques. That's why amyloid beta it forms plaques so easily. It's like it's a beta sheet machine, basically. And then lastly, the changes in the structural properties of the protein. Those can change based on the environment. pH and temperature, things like that, exposing a protein to a different environment like that can affect how the amino acid properties are going to respond, and it's going to affect if the protein gets denatured or unfolds, etc. It's very weird, these intrinsic protein properties and the hunt to figure out what exactly makes us susceptible to the disease and what if there's any way that we could stop the progression of the disease. It's all looking into the intrinsic properties of these proteins and these amino acids. So I mean, diseases diseases can help us understand what the heck is going on. So I'm gonna go through 3.5 prion diseases. We have Creutzfeldt-Jakob disease, which, as you can see, mainly affects these outer meatiness of the brain, among other areas. I'm going to talk about Kuru, which is another prion disease that mostly affects the cerebellum. These are in other animals, so Scrapia sheep, bovine spongiform encephalopathy is going to be cows, and then chronic wasting disease is more in deer elk type. So I care about humans. Lastly, I will be talking about the fatal familial insomnia, which is a prion disease that mostly affects the thalamus. So Creutzfeldt-Jakob disease. It is a very widely studied prion disease. It has it's just it I feel like it's like the textbook prion disease at this point. You can see on the right hand side there are abnormalities in this area, which I believe is the basal ganglia, which is why you get a lot of motor symptoms with this disease. Here we see abnormalities in the cortex. Looks like in the parietal and then sort of in the frontal areas as well as what's it called back here. This is part of the thalamus. So plaques build up in parts of the brain that have to do with consciousness, sleep-wake cycles, and motor areas. Generally, the cerebellum, which is sort of the hub of involuntary motor control, also gets affected. The symptoms are dementia, hallucinations, personality changes, myoclonus, which is involuntary jerky movements, and rigidity. So, and that's of course due to these specific areas of the brain that are affected. It's not shown on this MRI, but I'm pretty sure the hippocampus somehow gets affected too, because there's, I mean, dementia. There's a lot of memory lapses. Most cases of Creutzfeldt-Jakob disease are sporadic. So that's scary. I mean, it could just arise sporadically. 8% are genetic. Some people have a specific mutation in their prion protein that predisposes it to change into the infectious form. 5% are that are iatrogenic, which again are medical mishaps. So I think I have a. So the medical mishaps can be some people who need it's called a dural graft. It's tissue in the brain. Some people need a graft of that for whatever reason. Or I've also heard of if you need a corneal graft. You can accidentally be given tissue from someone who has a prion disease.
Ah.
So it's a medical fuck up, or yeah, it's messed up, man. And then 1% occur as variant Creutzfeldt-Jakob disease, which we will see in the next slide. That's what's referred to as human mad cow disease. So in the genetic version, FCJD or familial CJD, the most common variant is E200K. What that means is that a glutamate, denoted by E, is swapped out for a lysine, K, at position 200 in the PRP protein. Those are pretty different amino acids. Glutamate has a negative charge physiologically, and lysine carries a positive charge. So if you swap that, if you swap the charge, you could have a protein that interacts abnormally with itself or with other things in its environment. You can have disordered membrane interactions, so it will either it'll interact in a weird way with the cell membrane. And by weird, I guess I mean it interacts too much with it and could hurt it, or it doesn't interact enough with it and therefore doesn't carry out its function. Like normally membrane proteins have to do with signal transduction. So if it's not interacting with the membrane correctly, you're not getting the signal out. Or it could be it could changing the when you have like different charges in a protein, they are effective in different environments. So having a negative charge at a specific position, maybe it's more effective at this specific pH. But if you make it a positive charge, now at that same pH, it's not going to do jack shit. So just that one. It's one, you guys. It's one amino acid. It's just one. And it changes everything. So due to those changes, that makes it very susceptible, and I mean it's genetic, so it will at some point mutate and cause the prion disease in those individuals. These are the what I mentioned, the corneal growth hormone. I knew there was a third one. People who receive growth hormone implants from someone with a prion disease will contract that prion disease. And as Gray mentioned earlier, you can also get a prion disease from blood transfusions, I believe. Yeah, from blood transfusions. Another thing that I want to mention is before with the most of these cases involve that methionine or valine allele. I just want that to be kept in mind, the MV, because that's going to come up again. So CJD, Creutzfeldt-Jakob, it's associated with mad cow disease because humans who consume meat, tissue, whatever, from a cow that was suffering from bovine spongiform encephalopathy or mad cow disease will conduct variant Creutzfeldt-Jakob disease. Like Kuru, as you'll see, it is contracted when you ingest the prion. So it is different still. There's a reason why we have a distinction between the VCJD and CJD. And that is because the age of onset is going to be a lot younger for CJD. People with like the genetic or the sporadic CJD tend to be a lot older. Um, partially because as we get older, our body forgets how to make itself, and also because the incubation period of a prion infection tends to be very long. I love tentacle onion. In VCJD, there's not as much dementia symptoms, but more psychiatric and behavioral symptoms like heavier like personality changes, and maybe paranoia, things like that, but not so much the dementia. It's also from the point where someone is starting to have the symptoms of the disease, it is longer in VCJD. After the first bout of symptoms start, it's about a year until death. And for regular CJD, it's very short. It's like 5 months. It progresses a lot faster. Here's those MV again, the methionine valine 129. If you are heterozygous, because remember, we have one copy from mom and one copy from dad. If one allele is making it so that you have a methionine and then the other is making it so that you have a valine, it seems like there is some protection from it's so funny. There's some protection if you like eat BSE infected meat. There might be some protection for you from contracting VCJD. And it's just funny because this is like just not, you're gonna see this is not necessarily the case for prion diseases in general. So it's just kind of funny. Another difference between VCJD and regular classic CJD is how they look histologically, how the brain tissue looks. In CJD or in VCJD, we have these they're called florid plaques. I'm really not sure what is meant by florid. It might just have to do with the color of the staining, but they're these circular sort of the plaques are arranged in a circle, and as are these spongy sort of holes. This is not what classic CJD looks like. Did I have a yeah, classic CJD, there's no circles. They're not arranged in this sort of pattern, so it's interesting. They look completely different histologically. So, oh, and there's a really funny, guys, this is so funny. This wiki stem cracks me up. So it's from the CJD or VCJD page. In the US, the FDA has banned import of any donor sperm motivated by a risk of variant Creutzfeldt-Jakob disease, inhibiting the once popular import of Scandinavian sperm. That I just wanted to share that. I thought that was so funny. The once popular import of Scandinavian sperm into the United States. I just thought that was so funny. The wording of that is so good. Despite this, the scientific consensus is that the risk is negligible, as there is no evidence that Creutzfeldt-Jakob disease is sexually transmitted. Oh my god, the once popular import of Scandinavian sperm. I want to see the
source. I want to know when that is once popular.
I know, because this has
period of time.
This has an unheard of 3. Actually, that's probably for the no evidence that it's sexually transmitted. There is no citation. 29. Oh, it's 29.
No, 29 has it. Yeah.
All right, maybe
we gotta look. We gotta dig deep. I'm gonna go to the library and start looking at some fucking microfiches and shit.
Oh my god! Like, who is saying that this was popular?
The crates. Yes.
Like, exactly. Yeah. Oh my god.
Show me the crates.
Show me the crates.
Joe Biden.
Yeah, I thought that was the once popular import. Like you learn something new every day. So fantastic.
It's not for insemination. It's because people like the taste.
Oh my god. No, they don't. You guys eat lutefisk over there.
They like the taste, Rebecca.
Americans don't. Americans are normal.
We are not normal.
All right. All right. There is another prion disease that is caused by eating the infectious agent, and that is kuru. It is caused by funerary cannibalism. So what that means is there's this indigenous group in Papua New Guinea called the Four People, and one of their customs was after someone died, they would bury them and then exhume the body and actually consume parts of the body as a way to, I think, release its spirit. So it was something that indigenous group would practice, and at some point, this disease started to pop up just in these people, and it seemed to have to do with this practice of funerary cannibalism. So the I think that it was eventually accepted that at some point, around, I think this was around like 1900, a member of the Four People had contracted sporadic CJD. They had died, and then the people, of course, his body was consumed. So his infected with prion body was consumed by his family members, and they then developed this prion disease that did end up being actually pretty distinct from CJD, interestingly enough, because the symptoms had more to do with coordination and muscle movements. So there was tremors, ataxia, which is uncontrolled movements. Uncontrolled laughter was another aspect of the disease. And then dysphagia. The Blackfoot. Who are the Blackfoot? And I wonder, I mean, this disease, these types of diseases are really rare. So it's kind of like amazing and terrible that, you know, that this sporadic thing just happened within these people. Oh, Native Americans. Interesting. I mean, since the term funerary cannibalism was present in the articles I read, I assumed that it was a thing that probably more than just these people did, but I don't think it has ever, there hasn't been like a case of prion disease in many other groups that practice funerary cannibalism. So that's cool. I did not know that. Like, and it's specifically like it was in, I think, a specific family, but because they are of like intermarriage, a lot of other members of the group ended up contracting kuru. I think supposedly they don't practice this anymore. And the last person to die of kuru died in like 2005 or something. Because again, even though they supposedly stopped in like the 60s or 70s or something, the incubation can be crazy. It can be like a really, really long time. That is clearly not the case for this individual in the picture. This is the picture from Wiki. The ones in other articles were not as nice, but this is a very young person who is infected with the prion, who has kuru, and they cannot stand on their own. They don't have control of their muscles, and they're also very thin because they can't really eat. Dysphagia is trouble swallowing, so that tends to lead to a certain level of malnutrition. So yeah, both of these come from eating infected tissue, and yet they're ended up being pretty distinct diseases with different symptoms. We can even see in this chart right here. This is the amount of mutated prion protein in specific areas of the brain. We could see in VCJD it's got a nice spread. We're in the cortex, the basal ganglia, the thalamus, the cerebellum. It's kind of in a lot of different places. Kuru, not so much. Kuru, although it was found in those places, it was really, really found in a specific layer of the cerebellum, hence the more focus on those sort of disordered movement and disordered muscle movements versus CJD. Yes. Those are the exposure prion diseases rather than the sporadic, iatrogenic, or genetic. All right. This is gonna be some shit. I'm gonna get into my last prion disease, which is fatal familial insomnia. It is just as horrible as it sounds, if not more. And it has to do with this crazy weird mutation mechanism. Ugh. So, there's a mutation in both CJD and FFI called D178N, which is at position 178 in the protein. An aspartic acid is swapped out for an asparagine. One amino acid at this one place. Aspartic acid is negatively charged and it is hydrophilic. Asparagine is also hydrophilic, but it is polar and therefore neutral. It doesn't have a charge. So these are very different amino acids. Specifically, the mutation that occurs is a single nucleotide. We originally have GAC as our amino acid triplet. And that G, just the G, is changed to an A, and that's what makes it an asparagine. This is just this comes from the amino acid chart. It's not like a memorized thing. It's you gotta look for it. And those GAC always codes for an aspartic acid, and AAC always codes for an asparagine. So if you change that G into an A, it will become a different amino acid in the final protein. This is called a missense mutation. So it involves a mutation in the DNA that changes the amino acid. The incorrect amino acid is incorporated into the protein as a result of a missense mutation. We can call this an SNP, a single nucleotide polymorphism, which is basically what is important to, like when you're on 23andMe and you're looking at your raw data, it's going to be in the form of these SNPs, these one nucleotides, which we can also call, I'll throw the word allele in there. An allele is just a variation of a gene. So the normal allele here is D, the mutant allele here is N for aspartate and asparagine. This is crazy. This is just both of the diseases have this mutation, right? They both are associated with this change in the protein, in PRP. If on the same allele, you have a valine at position 129, you will get some form of CJD, whether it's genetic or sporadic, likely. If on that same allele, the allele that contains the 178 mutation, if on that same allele you have a methionine, you will get FFI. I'm so mad. I'm not mad. I'm not. But what the fuck? Not every case of CJD is going to involve this mutation. And I think the same goes for FFI. A different mutation that's not D178N could, I think, lead to FFI, but for some reason, with this mutation, it matters whether you have an M or a V on the mutant allele. And by mutant allele, I mean the one that contains this. Because you have two copies, and you are always going to be heterozygous for this. I can explain that in a little bit, but the other copy is not going to have this mutation. So the one that does, depending on if you have a V or an M here, you'll get Creutzfeldt-Jakob disease or fatal familial insomnia. I don't know which one's worse. They both are crazy. It is literally, yeah, it is a single point mutation. So silly. It's like, hello, this purine, bitch, not this purine. We want the G, not the A. This is blowing. This makes me so mad. But FFI, let's why don't we talk a little bit about FFI then? So that mutation, we're at fatal familial insomnia. So that mutation, the D178N, along with an M on that allele, for some reason, makes it so different from CJD. Because, I mean, the symptoms are, there are 4 stages of the yes, in any RNA transcription, because so on the DNA, this is gonna be AG, wait, purines, ATG, CG. Oh my god, I'm okay. I couldn't recover from the first one, and then it just got worse. CTG is what's going to be on your DNA. So, often what happens is we have the purine for purine, pyrimidine for pyrimidine. So that's what happened here. We have a purine for a purine. And that is hard into your genetic code. So anytime you transcribe it, the mRNA is always going to have that super, super wrong AAC. Because for whatever reason in the DNA, we swapped it out and we turned it into a T to make a TTG. It makes me so mad. It makes me so mad. And then beyond that, beyond just the change from the aspartate to the asparagine, what determines if you will get CJD or FFI is what amino acid is coded at 129. And that's a normal variation right there. All of us have either an M or a V at that position. And because we have 2 copies of that gene, you can either be MM, MV, or VV. So I don't know the amino acid code for valine, but methionine is AUG. I just know methionine. It's the start codon, so I know that it's AUG. Maybe we can pretend that methionine is GUG, or that valine is GUG. So again, GUG, yeah. So we have that what's it called, the purine switch again. But in this case, it's not a mutation. It's just it's a normal variation. It's an allele. You can either have M or V. So what further determines whether you will have FFI or CJD is that 129 M or V. It's crazy. So Kenzie.
Oh, same.
Oh my god. The best allelic combination. Holy shit.
Wow.
Fuck. But yeah, that really pisses me off. Like, ugh. We're talking about FFI though. So we have an M. That's what we're looking at the what? Oh. That's what we're looking at the protein right here. We have an asparagine here, so we're gonna have a prion disease. And we have an M here, so we're gonna have this one specifically. FFI is fucked up. There's 4 stages. First, you have insomnia. And then due to the insomnia, you get paranoid, you develop phobias, you are kind of going insane. Stage 2 is when you start hallucinating and also not sleeping. Stage 3, you literally do not sleep. Your sleep-wake cycles are only wake cycles. You have nothing for sleep. And you start losing a lot of weight. And then you get dementia, and then you die. It's fun. It's great. I mean, it's what is so interesting about FFI. As you can see here, CJD, you have that widespread spongiosis in the cortex, where you see the prion plaques and the degeneration. In Kuru, remember we saw that mostly in the cerebellum, which is at the top of the brainstem. In FFI, the atrophied cells are mainly in the thalamus and the entorhinal cortex. So the thalamus, I mean, this makes perfect sense. The thalamus has to do a lot to do with your sleep-wake cycles. It also regulates your consciousness, and it is basically a hub. We call it an integrating center, so it can receive signals and then disperse them to wherever they're supposed to go, basically. And then the entorhinal cortex, which I don't think it is not in this figure, but the entorhinal cortex is a place where the hippocampus communicates with the cortex. So it has to do with dreaming and memory consolidation and cognitive function. So again, this looks very different than CJD. It looks very different from Kuru. All because of this combination of letters, specifically. Oh my god. Furthermore, the different proteins that arise, whether we're looking at the FFI, which is what we're looking at, or if this is a V, it's going to be the CJD one. Depending on that, the protein is going to look different. The wild type protein, which is the normal protein, is going to look different from the FFI protein, which is the smallest, actually, of the proteins. It is 19 kilodaltons. And then CJD is going to look different from that, too. That protein is going to be a little bigger. It's 21 kilodaltons. They also differ in how resistant they are to enzymatic breakdown, and they differ in their glycosylation. The regular PRP protein is sometimes glycosylated, meaning that it is bound to this starch material that I believe helps it embed into the cell membrane. So when something is glycosylated, they are more resistant to breakdown by proteases. What?
I'm about to embed her membrane, if you know what I mean.
Stop it, you're gross. So what this translates to, basically, the type of protein being made, it's gonna have to do with how long the disease incubation is, the type and the spread of the brain lesions, aka is it going to be confined to the thalamus, or is it going to be more spread into the cortex? And then it's also going to determine the pattern of deposition in the brain. Is it going to be more like atrophy and lesions, or is it going to be more like spongy and like dead neurons and stuff like that? It is what the fuck? I mean, what the fuck? This chart shows the different glycosylation levels. So the far right, this dash right here is going to be pretty like unglycosylated, the naked protein. This one right here is going to be a little glycosylated. This one's going to be real starchy. So in both CJD and FFI, we have a lot of the glycosylated one, which makes sense because the protein is pretty resistant to being broken down. And then FFI, we have a lot more of the really glycosylated one and basically none of the unglycosylated form. So why is that important? I don't know. Something about this must make it so that the prion preferentially does its damage in the thalamus versus the rest of the cortex, like in the other prion diseases. What? I don't... Bitch. I love it. Remember how I showed you those, the names of all of those studies who were like, valine makes you more susceptible. No, methionine makes you more susceptible. Well, alleles matter. In this in this specific disease, at least, with this specific D178N mutation, did I? No. What the hell? Okay. You are more susceptible. Actually, I have to look at my notes for this because I want to make sure I say it right after having to go back and forth this, like, over what these different alleles mean. Okay. Let's just scroll. Oh my god. 1 and type 2. Dang it. Because this is important to say the correct one. Oh man, I really thought I had a definitive answer on this. How did I not write it here? All right. Well, I'm pretty sure that this huge study that I read postulated that being homozygous for the methionine made the disease a lot more severe, and made it shorter, meaning that you die faster. And if you were heterozygous, you died slower and had slightly different symptoms too. Like, I think one of them involved less of the psychiatric symptoms and more of the, like, I don't know. The takeaway is that this one mutation right here clearly makes the protein fold in a way that makes it susceptible to prion infection. And then this mutation's interaction with this one, so the asparagine interaction with the methionine, makes it does something to the protein to make it deposit in different areas of your brain. That is what I would really love to understand. It's pisses me off. And I had a lot of questions, obviously, at the terminal ends of my research on this. Obviously, I would love to know more about why the lesions are developing more in the thalamus in FFI versus CJD. Like, especially if one of the mutations is the exact same. It's so weird to think that one amino acid difference changes the protein in a way that makes it aggregate in different areas of your brain, or maybe it's that it's expressed in different areas of your brain. So in FFI, clearly the thalamus is like making and then fucking up more of this protein. I guess that would kind of make sense, especially considering that specifically in addition to the lesions in FFI, there's just really, really low metabolism in those areas. So it's just not even expending any energy. Jesus Christ. I would also love to know more about how the prion form, the PRPSC, induces a conformational change in the regular PRP. And if there's a mediator molecule. That was something that was sort of thrown around in the articles that I read, that maybe there is a protein X that also binds the normal protein and sort of forces it to change its conformation before having the misfoldedness occur. Messed up. And then other than just changing the sites for the proteases, which is kind of, I guess, my thought for the difference between the M and the V, is like maybe it just changes where the protein is cut, kind of like in Alzheimer's. But how does it actually change the shape of the protein, and how is it interacting with the disease mutations? Because all I really got was, oh, well, the N, it like hydrogen bonds with the M or the V. Like, okay, but that's like to chemistry. I need to understand how it affects the overall behavior of the protein. And as we were sort of discussing earlier with 23andMe, I definitely went and I like searched up all my stuff. And this is PRNP is the gene that encodes PRP. This is the name of the SNP. So all SNPs say, pretty much all SNPs say RS and then a lot of numbers. The build is the which build of the human genome they matched my stuff to. This is the position. These are the alleles. So at this position that you can either see an A or a G. And because it's a single nucleotide polymorphism, the what's it called? The allele is a nucleotide. So I'm heterozygous. It's just, we just look at this. These are the exact same. I'm heterozygous. Meaning that I have a methionine on one and a valine on the other. What I cross-reference my genotype with is a place called SNpedia. It's amazing. These people are doing God's work. So if we look over here, by the way, not every SMP, in fact, almost no SMPs have this much information for it. This just happens to be a heavily researched one. Like, people are really, really into prions. So what this is telling us is that if you have this genotype, if you're AA, then you have an increased chance of prion disease. And you don't take that at face value. It's just, it's genetics. It's just it's a susceptibility. It's a disposition. Because A codes for methionine. Because again, you know what? I'm actually gonna look it up really quick right now so that we can see. I want to see what valine's code is. I don't have them memorized because I never needed to. We're always allowed to have a chart. Okay, so yeah, GUG. Oh my god, I was right. I was pretending, but it's GUG.
Pay close attention to the guac chart.
Yeah. Actually, yeah, this is the guac chart. So methionine, the RNA code that codes for methionine is AUG. And the one that codes for valine, GUG. So it like I was saying, it's that purine switch. Adenine and guanine are both purines, and so sometimes there can be a mistake in replication or transcription or whatever, where oops, we put a different purine here. So in this case, though, we're not 100% talking about a mutation because this is just a polymorphism. It's just something that some people have this and some people have this, and it doesn't mean you're going to die or anything. It's just a difference. Some people have this. So this SNpedia is claiming, or whoever SNpedia is claiming someone's claim that the two methionines will increase your chance of prion disease. So this is why you don't take this at face value. Because, again, you saw that one mutation can cause one of two diseases. What if you don't even have that mutation at all? What if you don't have any of the other prion mutations? You just have this potential susceptibility genotype. Then you probably don't need to worry. This is more like if you have a sporadic mutation, like the D178N, then, ooh, and you have the AA, I don't know, buddy. If you're heterozygous, you have an A and a G, then you have resistance to the variant CJD. So if I ever accidentally eat mad cow disease, maybe I'll potentially be okay. I have less of a risk for the sporadic CJD, and I think that's because, again, the mutation. Right now, when I say the mutation, I'm talking D178N. The mutation, if it is on the same allele as an M, it's FFI, and if it is on the same allele as a V, it's CJD. That's the big important thing, because you have two copies of everything, except the germline mutation. You only have one copy of that. I think that might be a little that's what might be a little bit confusing. So my guess is the reason why heterozygotes would have a reduced risk of this sporadic one is because if I have a CJD mutation on my methionine allele, I'm fine. If I have it on my valine allele, I'm fucked, I think. This is where SMPedia kind of loses me a little bit. This person still has resistance. So that's a little different from what we were reading before. Because it seemed like either of these homozygotes would have some sort of susceptibility to the prion disease. And they even have a slightly reduced risk for the sporadic CJD. And then they just threw in an Alzheimer's here. Like, I think when you go on SMPedia, it will give you a magnitude variable. And the higher it is, the more you should look at this. It's kind of cool and important. And if it's a lower number, it either means that it's not important or that it's not as reputable. Like the sources are just kind of meh. And this was a pretty reputable one. Like, it was so, I guess I wasn't super surprised because this happens anytime I research anything, but just the back and forth between, is it this, is it this, is it this, is it this? It's very, very nuts. But yeah. Thank you, my friends, for listening. This was, I feel like I'm just going to end up saying this every time, but I'm going to say it anyway. This was one of my favorite things to research. It was so terrible reading about, like, stories of people with prion diseases, but luckily, because I'm on the molecular side, I'm mostly, like, removed from that.